Good evening, good evening. Welcome to the Koch Institute. Those of you who are visiting, we have many members of the Koch Institute community here tonight as well, including the Image Award winners, and congratulations to all of them. I'm really looking forward to their presentations to discuss the images that they have provided for the Image Gallery, and I, for one, think they're terrific. I'm really, really impressed. This is a third round of the Image Awards, and I think they are spectacular. The best collection of images that we've had to date, and I don't say that with every collection. Indeed, I think this is really a really exciting collection of images. So I'm Tyler Jacks. I'm the director of the Koch Institute. It's been an eventful day for us here in the Koch Institute. When I say that, I mean it's been a day full of events. We hosted the Koch Institute Leadership Council today, and many of my Leadership Council members are sitting in the audience, and it's wonderful to have you here. The Leadership Council has been tremendously supportive of the Institute since its inception and helps us do all the things that we do, and I'm incredibly indebted to them for today's advice and all the advice and support they've given me over the years. I'd also like to thank the judges who have adjudicated the Image Awards this year. Alex will give us the statistics about how many submissions we had, but we rely on experts to evaluate the art and the science that is represented in these images, and they do a wonderful job, and we appreciate their efforts. Some of those are in the audience as well. I also want to thank in absentia Charles and Ann Johnson, whose gift provided resources to build the space on the east side of our gallery, as well as a fund to allow us to change the art on a regular basis, to renew and refresh these exhibits, which is, of course, extremely important, and so we thank them as well. And finally, I'd like to thank members of our Within Sight Society benefactor group. This series that you're attending tonight, and many of you have attended Within Sight events previously as well, is an ongoing outreach from the Koch Institute to interested parties in our MIT community and our Cambridge community and beyond, to teach you a bit about what we do here, the science and technology behind it, and the impact that we hope to have in the fight against cancer. And tonight is an example of that. It's a very different style of event. We'll be hearing from the young people who've generated the images. But the Within Sight Society is a group of supporters of this activity. And we thank those of you who are current members, and we encourage others who might be interested in joining that group. And if you do join, you'll be given a wonderful collection of images from the gallery, shown by my assistant Carol Merrill over here. They are spectacularly beautiful, priceless images, one-of-a-kind images, and I think provide some incentive to become a member of that society, but of course the work that you're supporting is also extremely important. So I'm going to turn things over to Alex in 1 second, but let me just say from my perspective how excited I am that we do this, that we feature the science and technology that we perform in our laboratories in the way that we do out in the public gallery. It's actually quite unusual to show off the wonderful science and technology and discovery and invention that takes place behind the walls. To be honest, the reason that we first conceived of having a gallery space is that the city of Cambridge, which had to permit this building, Steve Mahler, who's sitting somewhere in the audience, is one of the architects, can attest to this. And the city said, you know, we don't want just a blank face of your building, and we don't want to just look into laboratories. We want it to be animated. And so working with designers, we came up with this concept of a gallery that would include these wonderful images and also other educational parts. And frankly, the more we thought about it, the more we liked the idea and invested in the idea. partly because we can tell the story of what's happening inside these walls in a really dramatic and exciting way that captures the attention of the people who are in the building and also are walking by. And we find people stopping and staring, and that's exactly what we wanted to do. We wanted it to be arresting. They would stop them in their tracks, and they would ask the question, what's behind that image? And they are strikingly beautiful, but the science behind them is even more interesting and more exciting, and that's what we really want to capture. So they can come in and read about the work. They can go on the web and see a video from one of these guys describing their work, and in that way learn and hopefully get excited to perhaps get involved in this quest that we have to do something important about cancer. So congratulations to all of you. That image happens to come from my laboratory. Lenny Gotcheva will be telling you about it. But I want you to know that I had nothing to do with the choices of the images that won in this round. But congratulations to all of you, and I think it's wonderful that we're representing your work in the way that we are. So I'll turn things over to Alex Fiorentino, who's our public outreach coordinator and organizes all of that and much more, including school tours to see what we do here. And I'll also take this opportunity to thank Alex for what is now years of service to the Koch Institute. I say it that way because I can't remember how many years. But Alex has been in this role for a while now, and he's done a terrific job. And Alex will be leaving us in June, I guess, to go off to medical school at Tufts, and we're very proud of him. I consider him a surrogate son of mine. Very proud of him, and I just want to thank you for all that you've done, Alex.
Thank you so much, Tyler, for getting us going and for the kind things you said about me and these scientists who have come to share with all of you what they do. So this is one of my very favorite nights of the year. You know, we have these images up on the wall year round, and I think we try to tell the stories as best we can on behalf of the scientists. But this is the only night when really the scientists are here all together to tell the story themselves and to share with you really what's behind the pretty picture. And so I'm really happy to be able to share that with you tonight. And the theme of the evening, in a way, is condensation, I guess, although that sounds like weather. But each of these scientists, each of these projects represents months or years of work, and we've given them the impossible task of describing it for all of you in 3 minutes. And in fact, the 10 images, or the 8 that you'll hear from the scientists tonight, represent only a small fraction of all the amazing science that's submitted to the awards. So I'd encourage you to check out our website. I'll share the link at the end of the night, but where you can look at all of the submissions to the contest and see not just the ones that were picked to be the most beautiful and the most interesting, but all the ones that didn't make the cut that are still really amazing science. But without further delay, I think we'll dive right in because we got a lot of talks to get through and a lot of great science to hear. So I'm going to introduce the first speaker for the night. Oh, sure, absolutely. So this is essentially a lightning talk. So each presenter will give only the 3-minute talk, and then at the end of the night, if you can hold your questions until then, at the end of the series of 8 talks, you'll have a chance to ask their questions. So keep them in mind if you can, and you'll have a chance to ask at the end. Okay, so our first speaker is Michael Wells. Michael comes to us from just across the street in the Department of Brain and Cognitive Sciences, where he works in the Feng laboratory. And his image is entitled Patch of Light, Trying to Understand a Newly Discovered Autism-Linked Gene. And I'll fade the light so we can see it a little better.
Hey, everybody. You guys hear me? Can you guys see me? I'm very short. I don't think this auditorium was built for Hobbit speeches, so I hope this works. So let me tell you a little something about this picture here. These are mature mouse glial cells. So for those of you who don't know what glial cells are, they are the support cells of the brain. So typically, when people think of brain cell, when they think of brain, they think of brain cells, they think of neurons. And neurons are the cells that transmit information from one brain region to the next. They would not be able to do that were it not for these cells. So if you have unhealthy glia cells, you have unhealthy neurons, you have an unhealthy brain. Okay, so this image was actually taken as part of a large autism study in which I was studying a protein called PATCH-D1, hence the title of this image. One of the first things you do, so PATCH-D1 I should say is a protein, a gene that's been linked to autism in human beings. So one of the first things you want to do when you're studying a new gene is to find out where it's expressed. So we're talking what brain region, we're talking what kind of cell, what cell types express this gene. So this is what this was. Essentially the way we do that is we throw antibodies directed against PATCH-D1 onto these glia cells, and we also throw antibodies directed against glial proteins. And if they overlap, which as you can see in this next image, which was not selected, when they overlap, you can see the two colors from each antibody. You can see the overlap. And so this data would indicate that PATCH-D1 is expressed in glial cells. We're still trying to iron out these details as to whether or not this is true, but if it is true, it's interesting because there are not many known autism-related genes that are expressed exclusively in glial cells. So the reason I picked the black and white image for submission, if you can go ahead and go back to it, I really don't know how else to put this, but I kind of fell in love with the darkness, and I know that sounds like I'm praising Satan right now, but that's not my intention. I really like the contrast and how this two-dimensional image kind of took on a three-dimensional character because of the shading and the shading pretty much does it. I can't take a lot of credit for this image because I feel like all I really did was used a $1 million microscope to take a picture of something that's already quite beautiful. So I love this image because it shows the inherent beauty of these cells, their morphology, how they're kind of out of control and they'll take over things if given the chance. So that's pretty much it for the science. I just want to say a few other things. First, I want to thank all my friends who came out here. I have a lot of friends in the audience who took time out of their day to, like that guy back there. I also want to thank my mom and sister who flew out here from Columbus, Ohio, where I'm from, to be here tonight. So on a serious note, I just want to say, this might just seem like a picture on a wall somewhere, but my family, we came from pretty much nothing. And now we're on display at the greatest university on the face of the earth. So thank you for that.
Thank you so much, Michael. That was terrific. So we'll keep things moving, and our next speaker is Lenny Gocheva, who comes from Tyler's lab here at the Koch Institute, and she'll tell you about her image entitled "Cancer Deconstructed: Investigating the Role of Non-Cancerous Cells in a Lung Tumor."
Thank you, Alex. Thank you all for being here tonight. So my main research interest is cancer, and particularly the area that I'm focused on is the tumor microenvironment. So what do I mean by that? For a very long time, cancer research has focused primarily on the tumor cells, and tumorigenesis was seen as a process where a tumor is a homogeneous mass of cancer cells that proliferate, invade, and migrate into the surrounding normal tissue, and that was really the whole story. However, what pathologists have known for many, many years is that when they look at a section of a tumor, they see an image that's quite different from this picture. So they see something more similar to this, where there's a wide variety of normal cells that are present within the tumor. So then the tumor microenvironment field is curious in knowing what are these other normal cells doing there. So one possibility is that they're just innocent bystanders and they're just there as a tumor developed. But as more and more research in this area has come out, it turns out that they can be very active players in the tumorigenic process. So for my research, I'm focusing on one of these cell types, and that's the fibroblast. And what I've done for this image is isolate two cell types. So these are the tumor cells here in red and the fibroblasts in green. And I've isolated these cells from a mouse lung tumor, from a mouse model that we have in the JAX lab that very closely recapitulates the human tumors. And what I'm aiming to figure out is how are these cells interacting. So the normal role of fibroblasts is very important. They're secreting a lot of factors that are building the scaffold of many of your organs. And they have a very important function during wound healing. So when you cut yourself, these cells in green become activated. Then they sense the stress and they start secreting factors that stimulate all the surrounding epithelial cells to divide in order to fill out the wound. Now, cancer cells are very smart, and they've figured out that these factors can be very beneficial for them to help a tumor grow. So they figured out a way to trick the fibroblasts into believing that they're healing a wound when what they're actually doing is helping the tumor grow. So using such a system in a culture dish, what we're trying to understand is this interaction. And now we know that this is more of a dialogue, so the tumor cells can talk to the fibroblasts, and fibroblasts in turn respond by secreting different factors. And what we're trying to do is listen in on their conversations and try to figure out what they're saying. But the first thing we need to do is actually learn their language. So it's really complicated, but systems like this allow us to gain more insight into what actually happens in a tumor. And the idea is that if we understand how these cells are interacting, we can come up with better cancer therapies, where we're targeting not only the tumor cells, but we're also targeting their whole supportive network, so that this gives us a better therapy for cancer. Thank you.
Thank you, Lenny. Our next image comes from Eric Williams, who works in the Guarenti Laboratory at the MIT Department of Biology, and it's also one of our extramural labs, so it's affiliated with the Koch Institute. And his image is entitled On the Scent, Investigating an Anti-Aging Gene Inside the Nose.
So this is an embryo that is 3 days old. At one point, everybody in this room looked like this embryo, whether you can believe it or not. This embryo has about between 30 and 50 cells. And one interesting feature of these cells is that they haven't made any decisions yet. And what I mean by that is as these cells divide and grow and the embryo develops into an adult organism, these cells have to make choices about whether they'll become different cell types, such as brain cells or skin cells, or lungs or stomach, muscle or bone, or a myriad other number of types of cells. And I'm interested in how these cells make these decisions. So this is the experiment. So I took an embryo, and I made an embryo that had both half red cells and half green cells. And I had this hypothesis that this sirtuin gene might be involved in making some of these decisions. So the green cells lacked this sirtuin gene, and the red cells had this sirtuin gene. So I let this embryo develop, and the idea is that as it develops into different tissue types, I can look and see if there's an equal contribution of red and green cells to that tissue, such as muscle, brain, bone, or whatever. And if I see that there's a lot more red cells or a lot more green cells, as opposed to a 50-50 mixture, then perhaps this sirtuin gene is involved in making some of these decisions. So I did that, and I looked at a lot of different tissues. I looked at lung, brain, muscle, and this is a picture of one of the tissues I looked at, which is the mouse olfactory system. So I'll use my nose as a way of showing what this is. So if I'm looking straight at you guys and you took a slice straight down here, you would see the inside of my nose. It would look different because this is a mouse. The mouse olfactory system is actually really quite beautiful compared to our olfactory system. But this is the center. This would be your septum. And these little spaces are where the odorants come in through the nose and bind to the neurons. The neurons are here outside, and you can see they're on both sides. And what I found is that, on average, there were more green cells than red cells. So my take-home message was that you need to turn off SIRT1 if you want to become an olfactory I really like this image because it has both symmetry and asymmetry. If you look at the left side, if you look at the structure of the left side and you look at the structure of the right side, the structure of the muscle factory system is symmetrical. On both sides, you can see the same features. But what's asymmetrical is the green and red signal. And it has to do with the nature of the experiment, injecting non-random population of red and green cells into different regions. So that's why there's a difference between red and green. But the opposing symmetry and asymmetry is what I really like about this image. Thank you.
Great. Thank you, Eric. Our next image comes from the Langer and Anderson labs up on the 6th floor of this building. The scientists who created it were Omid Vese, Joshua Doloff, Minglin Ma, Alan Chu, and Arturo Vegas. Omid is going to act as the representative of that group for the evening. And their image is entitled Sushi Implant, Seaweed Encapsulated Cells for Treating Diabetes. And this is what it looks like.
Thank you, Alex, and thank you all for coming tonight. Yeah, we have a quite expansive team upstairs, and we're doing some cool stuff that I wanted to share with you today. So one of the technologies that we're trying to develop is we're trying to develop an artificial pancreas. And the reason why we want to do this is as a way to treat juvenile diabetes. So for those in the audience that may not know, juvenile diabetes is actually an autoimmune disease, meaning the body's own immune system attacks the insulin-producing cells for reasons that are not quite well understood yet. So we have this idea that perhaps we can take these cells from healthy patients that normally produce insulin or from perhaps a stem cell source and protect them in a plastic-type matrix. And the challenge with this project is what type of matrix do we use, what type of plastic? So we have a big team and a robotics system where we're experimenting and evaluating hundreds of thousands, perhaps, of different types of plastics that we put these cells in. And the reason why it's called a sushi implant is because the basis of this plastic material that we use comes from seaweed. And it's a unique type of sugared plastic type molecule in that it can form spheres very well, which is the right type of format for this application. Spheres are good because they allow good exchange of nutrients, so insulin can get out and nutrients can get in. And this is actually what it looks like with these cells protected in this matrix. And what we have here is actually a prototype device that we actually put in a mouse, and we've taken it out after several weeks, and we've evaluated what types of cells are actually covering this matrix. And by doing these type of studies, we actually want to get to the point where we don't see any immune system cells on here, and those are all the beautiful blue and red that you see on the capsule. So this helps us understand what type of response we're getting, and by knowing that, we can address it and hopefully come up with better materials and a better device, which will ultimately hopefully be included in a clinical trial where we develop this technology. So thank you.
As an added note about Omid's image, I actually had a group of 6th graders from Norway, Maine here today, and they were dropping this same material into a solution and making balls that looked exactly like this. So that's another cool thing about this type of experiment is you can replicate it at home using kind of off-the-shelf materials. Anyway, beyond that aside, we'll move on to our next image, which comes from Tal Danina, Jeff Hasty, and Sangeeta Bhatia, from Sangeeta's lab up on the 4th floor here at the Koch Institute, and also Jeff Hasty is from the University of California at San Diego. And their image is entitled Bacterial Supernova, Programming E. coli to Release Drugs into a Tumor.
Okay, so there's a really exciting and emerging field of science called synthetic biology. And what synthetic biology tries to do is engineer new biological systems with DNA. So as you guys know, DNA is the language that encodes our genes and the life of all biological organisms on the planet. And for a while now, to understand biological systems, what we've done is add a gene, take out a gene, and see how this biological system behaves. But we're now at a point where DNA technology is so cheap and it's really easy to construct large pieces of DNA that we can manipulate entire bacterial systems all at once and even create entirely artificial bacteria. So the goal of this field is to create bacteria that produce fuels for us, sense and clean up environmental toxins, or treat disease. Now, to work with this amount of DNA, we literally have machines that print out DNA in whatever sequence we want. It requires an abstract language. So it's like if you wanted to work with a computer, to work with the hardware, you need a computer software. So to do that, we have an abstraction of this language called genetic circuits or genetic programs that we build. And these are elements of promoters, genes, transcription factors that interact with each other. And we can design these to produce a particular behavior. So I've been working with these systems in particular to make oscillators, which are rhythmic devices. And we've done this in bacteria using green fluorescent protein, and we can visualize single-cell oscillators. I actually designed a circuit that makes bacteria oscillate all together at once. And the way that we did this is by harnessing a bacterial system known as quorum sensing, where bacteria produce a small molecule that allows it to go through one bacteria and into another and relay its phase. So the image that you see out there is actually an experiment with that synchronized oscillator. And I was really interested in seeing how this pattern develops as a function of space and time. And I managed to get a single colony of bacterial stuck to the wall and capture this movie. That's pretty cool, right? So if you break it down, what happens is the bacteria reach a critical size where they've accumulated enough of this quorum sensing molecule. At that point, they produce a burst. You can see that only the bacteria with enough neighbors around them are producing this burst of fluorescent protein. And the ones in the center produce it first, and every other layer is a little bit slower. And as this colony grows outward, the intermediate layers burst again and again. Okay, so that's my image, and how does this relate to cancer? Basically, it's been known for a while that bacteria can actually home into tumor environments, and they like to grow within the tumor because they're sheltered from the immune system. The immune system is always chasing after them. So what we're trying to do now is take out this fluorescent protein and put a drug that bacteria can make so that they can kill the cancer cells from the inside out. And that's what we're working on now. Thank you.
Great. Thank you, Tal, for sharing that story with us. So our next image also comes from Tyler's lab. No nepotism here, but this is actually, I think this is the first 2 times this year that images from Tyler's lab have won, and I think it's completely deserved. They're both amazing images. So the image is entitled, switch to it so you can see it, "A Dangerous Meeting. Blood Vessels and a Tumor Come Together."
Thanks, Alex. Thanks, everyone, for coming out. So I'm Tuomas, and I'm a postdoc in Tyler's lab. And I'm interested in, as Lenny explained, how the cancer can use surrounding cells, in this case, cells that make up blood vessels, to reach a bigger size, to be able to actually gain nutrients and oxygen. So we know that cancers can grow beyond a very small size without securing their own supply of oxygen and nutrients. And you can imagine that choking this supply would really dramatically choke the tumor. I was interested in, when I took this image, I was interested in how lung cancers do this process. And to do that, I injected cancer cells that actually express that same green fluorescent protein that's originally isolated from jellyfish, so we can tell the cancer cells apart from the normal lung, and allow the cancer to form in this lung. And what we did after that was label the blood vessels of the lung with a red molecule. And what you can see here in the normal lung is a very dense vasculature. The lung, in fact, is one of the most densely vascularized organs of the body. And even though this is a still image, what you can maybe appreciate is that there's a lot of activity, almost movement, happening in this picture. You can see the cancer cells invading to the right, and the blood vessels actually invading to the left. this area here is actually part of the tumor but doesn't have any blood vessels. So these cancer cells here are suffering from hypoxia. They don't have oxygen. So that stimulates them to produce growth factors that actually attracts these blood vessels to grow and sprout towards the tumor. At the same time, the cancer cells are also invading the normal lung, which is already vascularized. Now, there's already drugs used in human patients for some types of cancers that block the process here, the growth of new blood vessels. But it turns out that a lot of cancers actually grow alongside previously pre-existing blood vessels. And this process is called vascular co-option. So instead of growing their own vascular supply, the cancer cells are actually crawling on previously pre-existing blood vessels. And this process, we don't understand that well currently. So learning more about this will definitely be a very important step in management of cancer. Thanks.
Thank you, Thomas. And actually, I don't think I introduced Thomas at the beginning by name, but his name is Thomas Tamala, and he's a postdoc in Tyler's lab. So our next image is presented in absentia because this scientist, Fernand Federici, as you can see, he works at Cambridge University in the UK. And actually, his image appears here at the Koch Institute as part of a partnership that we've set up with the Welcome Image Awards, which is based in the UK. And so each year we'll display one of their winners, and each year they will also display at least one of our images. Actually, this past year they displayed two of our winners. They liked them so much. So that's a great partnership that will continue in the future. And it allows us to display fascinating images like this one. So I'll give you just a very brief description. Similar to Thal's bacterial supernova image that you saw just a few minutes ago, Fernand is interested in synthetic biology, in kind of dissecting the components of normal biology and then using them as tools for engineering and applying them and combining them in interesting ways. Unlike Thal, however, Fernand here is looking at plant cells instead of bacterial cells. So we're looking at all the intricate components of a plant called Arabidopsis thaliana, one of the most commonly studied plants. And it's amazing to me just to see the complexity. You think of plants as being stationary and maybe boring, but there's so much complexity and interesting kind of machinery underneath the surface. That's really what stands out to me about this image. So our next image comes from the Heinz lab, also here at the Koch Institute, from Kwabena, Badu, and Kansa. And actually, this is the third image of Kwabena's to win the contest. So he's a very talented researcher, and we've been lucky to be able to display some of his work here. And his image is entitled, "Feel the Pulse, Smooth Muscle Cells Respond to Stretching Forces."
Thank you, Alex. So, as you said before, I'm Kwabena. I work in the Heinz lab, just in the third floor of this building. And half of our lab works in vascular biology, while the other half works in cancer biology. But one thing that we are all interested to some extent is how the external environment around the cells influence the way the cells behave. And what I like about this picture is that one of the things that you immediately get from it is all of these cells are aligned, almost as if they're responding to something collectively. And that's what's happening. I've exerted force on these cells, and these cells are aligning as a result of it. And so what we have labeled here is, so I dyed these cells with two chemicals, one that binds DNA, and so localizes to the nuclei, and one that binds a protein called actin. And actin forms long polymers that make up the skeleton of the cell, is what we call the actin cytoskeleton. These cells are called smooth muscle cells. And so to give you an idea of why we're doing this and how this helps us, I need to tell you a little bit of what smooth muscle cells are. So smooth muscle cells are cells that surround large arteries that emanate out of the heart. And so as the heart's beating blood, you can appreciate the fact that as a result of that, they're exposed to varying pressures and changing in force constantly in all of our bodies as we speak. And so that makes them a good model for looking at how cells respond to changes in force, because these guys are pretty much pros at doing that. And so what we're specifically interested in is how does the environment around the cells, a component of the environment that we call extracellular matrix, change the way these cells behave in general, and specifically in this case, how it changes the way they respond to force. And to give you an idea of what this extracellular matrix looks like, I have this picture, which I sent and didn't win. And it's the same similar pictures before in the sense we're looking at smooth muscle cells. And in violet, where you see it's a chemical that stains for actin cytoskeleton. In red, it's a nuclei. But in white is a protein called fibronectin. And that's a component of this extracellular matrix. So the questions that we want to answer are, what happens if you sever the ability of these cells to communicate with their outside environment? In this case, with fibronectin or other extracellular matrix proteins. How does severing that change the way these cells behave in general, how they respond to when forces are applied on them, or alternatively, how it changes the way they exert force on their environment? And so pictures like these are really important in helping us answer those questions. Thank you.
Thank you, Kwabena. So we have one more image creator, Rob Mathis, who unfortunately is traveling and couldn't make it tonight. He comes from the Gupta Laboratory, which is at the Whitehead Institute, and another lab that's affiliated with the Koch Institute as an extramural faculty lab. And Rob's image is entitled Molecular Watercolor, Mixing Fluorescent Proteins to Label Families of Cells. So Rob is doing something pretty unique here, at least from what I can tell. He's interested in cancer and interested in how the cells within a tumor can be very heterogeneous. Some of them are different from each other, and he's interested in identifying the cells within a tumor that are most dangerous, most likely to spread to other parts of the body and cause serious health problems. But what's unique is that he's not trying to identify the most dangerous cells here just by looking at them or characterizing their shape or what proteins they express. He's trying to identify the cells that are in different families. So there are many colors in this image, and each color represents a set of cells that descended from the same common ancestor. And so he actually he's looking at identifying which families, which sets of related cells are most dangerous and maybe would be best to target with drugs. And what's particularly interesting to me is the way he achieved all these different colors, many more than you would traditionally see in a micrograph. And it's actually the same principle that enables the display we're looking at right now to display many colors. And that is that you can produce a wide spectrum of colors by combining red, blue, and green in different amounts. And so he randomly, to assign these colors, randomly infected cells with different amounts of a red protein, a blue protein, and a green protein. And that randomness produced the wide spectrum of colors you see here. So, another very cool image. Okay, so moving on to actually our final image, and we're going in the order that they appear in the gallery. So this is the one on the far end of the west end of the gallery. And it was captured by Alex Shalik, who's here, and his colleagues in Hong Kong Park's lab at Harvard University and in the Broad Institute. And his image is entitled Cellular Injections, Using Nanowires to Investigate the Causes of Leukemia.
Thank you very much, Alex. My name is Alex Shalik from the Park Group at Harvard University. And first, I'd like to thank the Koch Institute for agreeing to display our image. The image in and of itself is a scanning electron micrograph of B cells on top of vertical silicon nanowires. And just to provide a little context for this, because this is maybe one of the simpler images you'll see tonight, in the Park Group, we're interested in using cutting-edge advances in micro and nanotechnology to study biological systems. Essentially, what we're trying to do is create the doctor's toolkit for studying cells. So if you think about the tools that a doctor would use to study your body, you think about things that are maybe a little bigger than you, say an operating room, to things that are a couple of orders of magnitude smaller, like a needle. By analogy, an average cell is about 10 microns in diameter. So what you want are things that are tens of microns, say a microfluidic device, in case you've come across that term, or things that are tens of nanometers, like a nanoneedle. And that's what we have here. Basically, we have a syringe for injecting things into cells. And it turns out that if you optimize this surface and you play with the surface chemistry, you can penetrate the membranes of these cells in a minimally invasive fashion and deliver just about anything you want into these cells. And so what we've been doing in the Park Group over the past few years is using this technique to study the molecular circuits that drive cellular function and dysfunction in healthy and diseased states, and to try and figure out better therapeutic avenues for studying these cells. And this in particular is one representative image of our work with immune cells, of which we've done a lot, and if you have interest, I'd be happy to tell you more about it offline. Thank you very much.
Okay, thank you, Alex. So that wraps up the talks, and now, as promised, we'll go into a Q&A session. So I'd like to ask all of the speakers to just wrap around up here. And I'll, just in case you forgot, I'll put all their images up on the screen. And Sarah, my colleague, is in the back of the room with a microphone. I've got one up here, and I will give one to the speakers. You guys can just pass this. Does anyone have any questions about these images?
I'm going to tell by my question that I'm not a scientist. What do you guys think about Jackson Pollock now? Maybe I should rephrase that. How do you feel about abstract expressionism?
I think we get to appreciate his work even more now.
Okay. Who else? Anyone else with a question? I saw another. At least one more.
Hi. Hello. This is for Lena. Is that your name? I was fascinated by this picture, but I was really fascinated because I think anaphase is going on in one of your nuclei. And I was wondering if that was something of interest to you to see how cell cycle is progressing within these cells.
You mean within the tumor cells?
Yeah.
Yeah. So of course that's a process we're very interested in studying because.
See, there it is. It's all lining up inside that nucleus.
Yep. So it's very cool because in culture system like these, we can actually catch exactly the processes that we see that happen in vivo. So we can see cells dividing. We can even do live imaging to watch them, how that happens. And using these co-culture systems where we have multiple cell types mixed in together, we can see how these interaction can affect these processes. So we can test it with the stromal cells or without and see how the processes are involved.
Are you going to go on to a 3D system, like form a gel and see how the extracellular matrix is interacting with all of this?
Yeah, so from this, the next step is a 3D culture with matrix to more accurately mimic the in vivo system. And then, of course, the ultimate test and the ultimate system where we want to study this is in a whole organism. So that's why in the JAX lab we have some very sophisticated mouse models of cancer where we can, again, study the same processes and using what we've learned from simpler in vitro systems, then also take them in vivo and even test different drugs, etc., as a more representative situation.
Thank you. All of your images are beautiful, by the way.
Josh?
Hi, my question is actually for Thomas. So you're using, I guess, a fluorescent protein to label tumor cells and then watching how they interact with the blood vessels in the lung. And since this protein in particular is not from a human, I know in particular resident immune populations such as macrophages are important for tumor blood vessel formation and infiltration into a tumor mass. So have you looked at whether or not this foreign protein is affecting the resident immune populations and whether or not what you're seeing here is in fact what actually happens when you have a normal tumor and a normal lung?
That's a really, really good question. I didn't explain the experiment in such detail that would have answered it preemptively. But so what I did in the first experiment, that is actually the image, was implant these GFP, these fluorescent protein positive cancer cells into an immune deficient mouse. that lacks T cells completely. So the tumor cells are not rejected, nor does the immune system react to that very, very foreign protein that, like you say, comes from a jellyfish. The next image that I showed, however, was from an actually more advanced model of cancer that Tyler has pioneered in his lab, where tumors actually start from a single cell within an immune competent mouse. And this image up here, this actually will be a much more accurate model in studying what happens in the human. And this image actually is taken from a tumor that started forming only 4 weeks, for only 4 weeks. So it's a very early time point.
I just want to first thank all you guys for all your beautiful images. It's really great, all the science you guys are doing. I really appreciate all your hard work. My question actually was for Michael Wells, who had the kind of top right image in black and white. And so we've heard a lot tonight about cancer biology and that sort of thing. And but I was struck by Michael's image because he mentioned that had to do with autism research. So I'm just kind of wondering, how do you feel about autism research, how it's sort of progressing in relation to cancer research, and specifically sort of how autism research is, how you're tackling that, and what's the sense in the field of the best way to look at autism, and how are you particularly looking at autism when it's such a debilitating disorder?
So I can't compare them too well just because I'm not exactly up to date with what's going on in the cancer field. But in terms of how we're looking at autism, at least on my end, is through the use of knockout mice. These are genetically engineered mice. I think somebody mentioned this concept earlier, this idea of removing a gene and assessing its effect. So, like I mentioned before, I'm studying an autism-related gene, and so what we have done is remove that gene from a mouse with the idea of assessing its behaviors, see if they're autistic-like, and the long-term goal of using either pharmaceutical intervention or gene therapy in the long term. So I hope that answers your question. I know nobody's asked this yet, but just to get this over with, nobody's asked me who I'm wearing. This is Calvin Klein, so I was waiting for that question. We don't have much time left, so.
Thank you for reading my mind, Michael.
Thank you. No problem.
Any other questions? Okay, I see one back here.
This is for Alex Shalik, which was the last image. And I was just wondering, how did you make the needles?
So there are a number of different ways that we do it, but the simplest way of doing it now is actually to spin a resist on top of a surface and then to use a gas to etch down the surrounding material. So basically you're protecting one spot and turning what's originally a spot into a cylinder. And as it pushes down, it turns into a wire.
What kind of materials is that silicone? Or?
Yeah, so this particular set's made on a silicon wafer. The outside of them becomes covered with a native oxide, so it's glass on silicon.
Okay, any other questions? Okay. If not, then I just want to thank all of our winners for not only all their great work that they've done as researchers, but also taking time out to kind of talk to all of us and help communicate what they do. I think that's a really important part of what we're trying to accomplish here at the Koch Institute is to really talk about science and engage people in science and take it out of the black box and into something that people can approach. And I think all the speakers tonight have done an excellent job of doing that. So if we could just give them all one last hand. And you can't literally take them home with you unless you happen to be one of their wives or husbands or boyfriends or girlfriends, which I know there are some in the audience. But for the rest of us, I'd recommend a couple of other ways if you want to learn more. One thing you can do is log on to our website. So we have a website for the galleries that shows not only their images, but also all the ones that didn't win. And for them in particular, tomorrow, if you can wait until tomorrow, there will be videos posted of each of them explaining their image, and you can watch that, share it. It'll also be on YouTube, and so I'd encourage you to check that out. Here's a sampling of some images from last year, from 2012, that didn't win, and in fact there are over 100 more images than this that didn't win last year that are equally striking. So I'd like to thank you all for coming out and joining us. And one more thing I'll mention is that if you enjoyed tonight and enjoyed the With Insight series, I'd encourage you to come back. This will be continuing throughout 2013. So our next event will take place on May 23rd, and it will feature, not like this, it won't be a night that features the images of the gallery. Instead, it will feature a particular thread of research and 3 different expert perspectives. So we'll have our own Professor Sangeeta Bhatia talking about very high-tech, exciting innovations for detecting cancer, and she'll be joined by a physician and others who can talk to this challenge and exciting ways that it's being faced. So thank you all, and have a good night.